Thank you, Margaret. I appreciate this invitation, Paul and Pete. This is fun. I like the opportunity to talk to folks here in New Mexico. I get to talk to high schools from time to time, and we've done. You just said that you do this at the bar sometime in a pub. That would be cool too. Yeah. So I'm going to talk about this weird journey we've been in over the last 5 years or so. I have a career studying air pollution, and in the air we have these particulates, these little dusts, and it gives me sort of a unique perspective from a lot of other toxicologists and a lot of material scientists and stuff, because particles are not so much chemicals when we think about chemicals in the body like dopamine or oxytocin or any of these weird things. Those are the bulk of what people study in science, in biomedical science, and they don't think about little tiny particles of stuff. And so we dove into this about 5 or 6 years ago because what was being done scientifically was sort of immature, just to not be too negative about it. And what we found is, like, every time we scratch the surface, we uncover something a little bit more, like, I hate to use the word alarming, but certainly compelling, that something is going on that's a little off. I always feel obliged to start with a financial disclosure. All of my grant funding comes from the National Institute of Health, and I am a scientific advisor for Circulate Health, which won't enter into any of this, but just to be on the level. So I'm going to divide this up into sort of chapters. First is just an overview of the problems. And this image was taken by a postdoctoral fellow in my lab. And y'all can hear me okay, right? I don't need to stay focused on... Oh, barely? Okay. I can stay a little hovering over the microphone at least. It's sort of early on a Saturday to be, like, enunciating to the back of the room. So we came up with this idea to capture real environmental plastics from the ocean, and rather than, like, go out on a boat, there's this beach on the southern point of the big island of Hawaii that collects this, like, tons of plastics every month, and the community group has to go out and clean it up. And I sent my team to help, and also to bring some stuff back that we could use in a research lab. And what they found was this pipette tip. So that little thing he's holding is just a universal tool in the laboratory, and like across the planet, people use these little things and throw them away all the time. And I have no idea how one of those would ever get into the ocean. They should have been thrown away, probably incinerated because of a biohazard, whatever. But there it is. But the other part of that is we know that the things we do in our world are contributing to this, and I don't consider any of this to be an us versus them problem. At the pub, I'd be happy to throw down on petrochemical industry. But for the most part, they're people earning a living, they're smart chemists, I knew them in grad school. We're all part of this, and so it's really something we have to solve together. One of the nice things about the research that we've been doing is it has caused a big surge of awareness. 80% of Americans think that microplastics are a problem now. So I feel like this is a great opportunity. When Richard Nixon was in office, there was such bad pollution, both water and air, that he created the Environmental Protection Agency. That was not something you would have expected from his administration. He signed into law the Clean Air Act, the Clean Water Act. What it took to get us there was horrific pollution. The Cuyahoga River in Ohio was set on fire, and we had The Silent Spring written by Rachel Carson. But we're in a moment now which is very much akin to that, and so it takes a little bit of patience to get through, but we do feel like things are moving in the right direction. So my story starts with a science fair project for a 7th grader. This is my kid Sam, back in 2019. He's now at Oregon State studying chemical engineering, and I hope he uses it for good, not evil. But it's an excellent discipline. Could go either way. But what we decided was, okay, there's a lot of papers coming out on microplastics, and let's go see for ourselves if these are really in our environment. So we went up to essentially the headwaters of the Rio Grande. If you've been up past Creede, Colorado, there's this reservoir. It's absolutely beautiful. The Rocky Mountains are spectacular. So this map, there's these little shaded blue areas, and the little tiny one upstream of that dot is all the watershed upstream from that one point. And only about 700 people live in and around that area and contribute. And then we sampled again at Taos, and now there's that darker blue area that's the San Luis Valley, about 80,000 people, probably twice that many cattle. But there's more and more impact from human activities at Taos. And then we get down to south of Albuquerque next to Isleta Pueblo. And so we sampled at each of these points, and then using 7th grade science, I mean, I got him a little filter and a little black light and a microscope, so he had help from Dad for sure. But what you could see was very obviously, as there's increasing human impact, there's increasing plastics that you can easily visualize under a microscope. That was another thing that bothered me the most about this study. What bothered me was how easy it was to find plastics in the headwaters of the Rio Grande. Like before you even have a chance to use that for irrigation, it's contaminated. So that's led us down this path and trying to understand the problem a little deeper. The issue is all the plastics, the lid of this reusable mug will eventually get old and break and be thrown away and then break down more and more. And it'll go from macroplastic to microplastic. When you talk about microplastic, you're talking about sizes from 1 micrometer to 5 millimeters, up to about a grain of rice down to a little bit smaller than the nuclei of your cells. But then it'll break down further and it'll become nanoplastic. Now, nanoplastic is something, it's really challenging to measure. The microscopes we use to detect microplastics don't work for the nano. We're at this new juncture in the field where we're really trying to understand the nanoscale better, because what we see and what we're hoping to publish soon is that the size of these things is really the size of viruses. And so they can be in cells, around cells. How they're handled is something that's really unknown right now. As Margaret alluded to, we used to not have this problem. Everybody in this room, I suspect, was born without plastics in their brain. I can guarantee you that is not the case anymore for children, maybe anywhere in the world. We definitely see that plastic particles are in the placenta and in the brains of the fetal tissues that we've been able to look at. So we've been producing exponentially increasing amounts of plastic since the 1950s. It's not a log 10 scale, it's a log 2 scale, but still doubling every 12 years or so. Right now we produce, I think last year, about 450 million tons of plastic around the world. In the next 8 years, we anticipate that should be 600 to 700 million tons. And it just keeps going up. And I like this quote: anyone who believes exponential growth can go on forever is either a madman or an economist. Apologies to any economists in the room. So to go from these fresh plastics, though, to the microplastics and nanoplastics, depending on the polymer, could take decades, if not 100 years. So think about, I like to use the example of the dashboard of your car. You buy a new car, maybe you're really good and you drive it for 10 years, 15 years before you decide it's time to get a new one. Well, then it gets sold to somebody else and they drive it for 5 or 10 years. Eventually it just doesn't work anymore and it gets junked. Goes to a lot. It's, drive down South Albuquerque and look at all those wrecker yards. They're still full of cars, and the dashboards are, you can scrape them, they're falling apart, but they haven't fallen apart, you know what I mean? So the time from this to becoming microplastics could be 2 or 3 generations of humans. So if we wanted to change something now, it's going to be our grandkids and our great-grandkids before we start realizing a true impact of this. So this is what gets me worried. And I will tell you, I'm not so worried about the health impact of these things right now. It's the unknowns about the future that bother me. Right now there's over a ton of plastic waste for every human on Earth, probably closer to 1.4 or 1.5 at this point. This is an old slide. And I love these Sankey plots because on the left side you're talking about how much total stuff there is and where it goes. So this is a little old. It says 8 billion tons of plastic. Most of it, over 4 billion tons, has already just been thrown away, already turning into micro and nanoplastics. About a quarter of it is still in use. Again, it's this cup, it's the dashboard of your car, it's the plastic bags. I'm just going to walk over and point. This cute little green line is how much we recycle. It's about 10%. I think our mental state is like, once you've recycled it, it's good. Everything's happy and everything's good. But notice this other little curlicue. That's the amount that gets recycled twice. So now you're talking about 1% of plastics get recycled twice. When we say recycle, we tend to think like it's forever, but it's really not. So this is a wasted kind of thing here, and we spend so much money and energy on recycling. And I always get into the state of like, please keep recycling, we gotta try, but it's a lie. It's a big lie that has been told to us for a long, long time now, and we actually believe it. All right, this is the big myth that I would like to share with y'all, because I know that the media gives you this idea that, oh, the fresh plastic, the wrapping of the food and the plastic water bottles, you drink it and those are giving you plastic. That's not the real problem. And I get a lot of grief from other plastics advocacy groups. Beyond Plastics, the advocacy group, invited me to give a talk and then saw that I was going to say what I'm about to say, and then they were like, we don't want you to say that, so we're not going to have you give a talk now. The problem is we're going to throw that bottle away. And so instead of a few microplastics coming off of that bottle, and I promise you, you can take a water bottle, empty it, and refill it 10,000 times, and you won't be able to measure the change of weight of that bottle. But eventually you throw it away, and the entire mass of that bottle is going to become old and decrepit and turn into micro and nanoplastics. So we fundamentally have this story backwards. It is good to try to change people's behavior now, but it's not the real part of the story that's concerning. All right, so what is the evidence that plastics are causing harm to humans right now? The evidence is not strong. No matter how big a paper you've written, there's just not great evidence right now. The problem is it's a new scientific field. Richard Thompson from the University of Plymouth in the UK discovered plastics in 2004. It's only about 22 years ago. And for about 20 years, the field was exclusively in the zone of ecologists, oceanographers and soil and water people. And they didn't really care about the nano stuff. They didn't study the nano stuff. Bless their hearts. What I'm finding now is that 20 years of science doesn't really tell me much about what goes into the human body. 2021 was when we first had a paper from a group in Italy showing, I think these are plastics in the human placenta. Does anybody else see this? Since then, 5 years later, a lot of us are going, yeah, I see that. And now we're developing better methods to do that. So the assays, though, are new. Doesn't mean everybody likes your assays. I promise you mentioned the April 8 article. Go back to January in The Guardian, and there's this hate-filled, scathing article about our paper, and it's like, well, I mean, you do it better if you don't like it. But there's a lack of consensus around what are the best methods to use. I will argue the skepticism remains warranted for the papers that have been put out, including my own. However, we're moving this field dramatically, rapidly, and in fact, 3 days ago there was a paper from China that, in my mind, more or less completely corroborates the work we have done. It's very nice to see it. What are the major, so I'm gonna review some of the big papers, but also just give you a hint as to how this gets into our bodies and how it moves around and what we think might be bad. I want to point out, I'm talking about the little plastic particles. There's also plasticizing chemicals, and that is a whole different field. There's a lot more known about it. It's a lot easier chemically to say, oh, I'm looking at this one particular phthalate or this one particular bisphenol. Easier to do. much better, robust understanding of how those things impact human health. So I'm not throwing them under the bus. There's definitely something about plasticizing chemicals that's concerning. All right. So March of 2024, this really cool paper came out in New England Journal. My group was starting to publish what I thought were cool studies, and then this group from Italy, Giuseppe Pauliso and Raffaele Marfella, came out with this, and I was like, oh man, you guys, this is really good. So among the things that they showed was that if you simply divide your patients into haves and have-nots, what they did was they're cardiac surgeons, vascular surgeons, and so there's a carotid surgery to remove plaques from the carotid artery, and so they used that tissue and measured the amount of plastics in that. And some patients had detectable levels, some people were below detectable levels, and they just divided it and said, okay, what happens to those individuals over the next 3 years? And what they found was, you see in red, that patients who had the micro and nanoplastics had a 4.5-fold increased risk of a subsequent cardiac event or a heart attack. That's more increased risk than any cholesterol-lowering drug is going to prevent, just to put that in perspective. So, but it's an associative study is the problem. So there were a lot of concerns expressed. One is about the method they use to detect, and it's a method that my group uses as well. And I will admit that the levels they were showing in these were absurdly high, but we think it's proportional and analogous, so we don't lose our minds about the concentrations in these studies. But the associative nature, it could simply be that there were people who had worse disease, and as a result of the inflammation or whatever, more plastics get taken up into that plaque. It's not that they cause the disease. So we have to interpret that cautiously. And when I get to my paper, you need to be thinking the same thing. All right, so that paper came out, and we're like, well, you know, there's this, and there's a lot of rat studies and mouse studies where people are looking at purpose exposures. You know, I gave a mouse a bunch of polystyrene microspheres or polypropylene, and things got worse. But we didn't know what the right doses were. We didn't know what the right route of exposure was, and we didn't know what the right materials, the polymer type and the size and shape, all that stuff is sort of a mystery. So my group really focused on trying to understand all of that better, especially in human studies. And I like these histological images because they're sort of oddly pretty. This is a kidney from a autopsy specimen, and the little glowing things we believe are plastics. But this is it's a region called the glomerulus. You see this circle. So this is where the blood comes into the kidney and interfaces and gets filtered. And you can see that you get a lot of plastics in and around those glomerular regions. And it kind of tells us that the kidneys are trying to do their best. They're actually trying to clear this, which, you know, it's one of the more optimistic parts of the study. So like I said, oceanographers, they've been looking at big particles that aren't really relevant to humans and what they can absorb. So we realized we needed to come up with better methods in this PI-GCMS, pyrolysis gas chromatography mass spectrometry. I won't burden you with this. I know everybody studied up on organic chemistry before they came in, right? So it's essentially giving us a chance to sort of collect all of the plastic particles and measure them at once as a cumulative mass. So it doesn't tell us everything we want to know, but all these other spectroscopic methods, Fourier transform, infrared, Raman, etc., they won't let you really look at anything smaller than 5 micrometers. So the really small nanoscale stuff we thought was important would not be detectable with those methods. So we came up with this new method. Essentially what we do, maybe it's inspired by being in Albuquerque. If you've seen Breaking Bad, you know how you can get rid of a human body, right? You just put it in a barrel full of chemical. So we can take specimens, and we worked with the Office of the Medical Investigator. They were wonderful partners in this. And we put small pieces of tissue into glass jars full of potassium hydroxide. It's base, very strong, powerful base, and it digests the tissues. You can also use a strong acid. You can use hydrogen peroxide at a high concentration. There's all kinds of ways to do this. We chose the one we like best. We let that sit for 3 days. We then spin really hard in an ultracentrifuge, and what that does is all the solid particles come to the bottom and pellet. They become a little solid mass at the bottom. And then we put that in our gas chromatography mass spectrometer, and it sets them on fire. And the gases that come out of that we identify as having come from burning polymer. And so it's nice. So we can look specifically at the gases that come from polystyrene, polypropylene. I took out the details. That's probably for the best. But we can look at 12 different polymers. What we see most is polyethylene, polypropylene, polyvinyl chloride, nylon, and let's say styrene butadiene rubber, which is tire wear. So the tires that are driving by all the time are just shedding little bits of plastic particles. We think of tires as rubber. It's a synthetic rubber. And they definitely leave potent bits of toxicants all over the place. It's much worse if you're a salmon, just saying. All right. So the first thing we did with this was we followed up on Antonio Ragusa's placenta study, and we thought, well, he was only able to look at really, really large particles when he published 5 years ago. So let's apply this method. And this is Marcus Garcia. I've got this wrong now. He's now assistant professor, so his career's been really moving along nicely. But we had 60 or 70 placenta samples from women who gave birth in Houston, because there was a group at Baylor who had assembled this repository of tissues. And this graph is essentially, each one of these bars is a different placenta, and the different colors represent the different types of polymer that we showed. And then you can see the summation of that on that chart. So most everything that we're seeing is polyethylene, polyvinyl chloride, nylon, PET. I keep forgetting about PET. PET is an interesting one because that's your drinking bottles, right? It's really surged since the 1990s in the marketplace, and now we're starting to see that trend in the human body. So this was interesting. We show really low levels in some people, but some people have extremely high levels, which was a little surprising. The next paper we did, this was sort of strategic on our part. We looked at human testes. And this is John Yu. He's in the College of Nursing. He's a reproductive toxicologist. And he'd already been doing a lot of studies related to toxic chemicals and sperm production using the testes from dogs in and around the Albuquerque area. So he has this cool connection with the veterinary clinics around town and with the humane the shelters. So he's able to collect these and sample the sperm and do the tissue analysis. And we worked with the Office of the Medical Investigator to get the autopsy testicles as well. And what we found was that humans had about 3 times the amount of plastics in their testes compared to dogs, even though dogs kind of eat off the floor and you'd think that they're getting worse things in their diet. The other thing I find fascinating about this is this graph here. This is the amount of polymer over age. So I'm just reading the tea leaves in this data, but if you notice, it seems to be higher in men of reproductive age, when the metabolism of the testes is going to be the highest, and it wanes in older age. The graph on the left is from another paper where they show that explicitly, like this is the curve you get of metabolism in male testes. So it really gives us some clues as to how the plastics are getting into the body. Maybe they're not just passively coming in. Maybe there's some active lure or mechanism that's pulling them into the body if you need, I'll get to this eventually, they tend to be sneaking in with the fats of our diet. And so if you need fats for energy, these are coming in potentially as needed. All right, and then that got us to the strategic part of this was the first environmental paper I've seen in a long time that got tweets from Joe Rogan and Elon Musk. So with this paper, we're starting to cross some barriers that I thought was really important. We then published this. So this was in Nature Medicine last year. This caused a tremendous amount of hubbub. essentially what we did, working with the Office of the Medical Investigator, we got 20 to 30 decedent samples of brain, liver, and kidney, and we assessed samples who died in 2016 and compared them with 2024. And the upshot here is that the liver and the brain showed an increasing trend over that 8-year period. The brain was about 40% higher over 8 years on average. Now there's a lot of noise in the data. We beat ourselves up around this technique of pyrolysis GC because it does seem very noisy, and I liken it to looking through a very blurry telescope. Like I can use that telescope, I can definitely see the sun with it, I can see the moon, I can kind of make out Mars. You know, it's not that great of a telescope. But the irony is when I do mouse samples, laboratory mouse samples, it's really consistent. So I think there truly is some variability among humans. If you're a vegetarian, if you just have genetic makeup that makes you clear it or take it up different, I think there's really something going on. Anyway, the upshot is plastics are in the brain. When this was reviewed, the reviewers, the blinded reviewers said, well, you need to show us more data across a wider range of time. I don't know if any of you have tried to find brains. It's harder than you think. And in fact, there was a big oops from the Smithsonian Museum a couple decades ago where they were like, wait, one of our investigators has just been sort of collecting brains from indigenous children for the past few decades without consent. So as a result of that, there's now a treatise that really stops museum collections from donating to labs like mine that are going to use destructive analyses. So we set things on fire, right? There's no way around. You're not getting your brains back. So we've had a lot of trouble trying to procure anything. But fortunately, a colleague at Duke had reached out to me, Andy West, and he had all these samples from patients from 1997 to 2013, admittedly from the East Coast, so there's some differences, but they were all universally lower, and the overall slope, that 40% increase over 8 years, doesn't change a bit. And then the reviewers also asked us to look at dementia brains, and I still think this is a strategic mistake because it causes way more alarm than it should at this point, but they were much higher. I'm going to skip this. This is fun though. Laurisa Barella has been looking at one brain from a donor from the OMI, and she's taken like 350 little spots and assessed it. So we can now look more completely at where these are distributing in the brain. And you see the redder areas is where there's higher concentrations, and it tends to be in the frontal cortex, where we do a lot of executive thinking. What else is important about the frontal cortex? High metabolism. So again, it comes back to that story of maybe there's something pulling them in. The brain is your most metabolic organ, right? When blood leaves your left ventricle of the heart, it goes to every organ of the body. But does anybody know what percentage of the blood goes to the brain? 30%. I mean, there's one little tiny spot gets 30% of the blood flow. So yeah, it demands a lot. All right, it was nice to do that study where we quantified the plastics, but it didn't tell us a lot about where the plastics were in the brain. So we developed this method called polarization wave imaging, and there's a refractile or birefringent property of many plastics. So it bends light and it organizes light. So if you put a filter just right on this, these things pop out. And I'll go back and do this again because this is an immune cell in the brain, and this is an immune cell. And these little dark spots are probably bits of dead cells that they've eaten up. And what's interesting is like, that's not all of the plastics. So the immune cells are there, but they're not necessarily responding to the plastics. They're all around them. It's really, this is all kind of cool. We found in normal brains, it was actually quite hard to find the plastics, even though we showed that there was really, really high levels there. They were all universally nanoscale. The best scale I have here is this is a nucleus from a cell, and that should be about 5 micrometers across. And here's a relatively big one, but it's a cluster of a bunch of particles. And then down there you can see they're all very, very small. When we looked at the dementia brains, it was very obvious. It was very easy to find these plastic particles. They were mostly in and around the regions of inflammation and degeneration in the dementia cases, but also along the vascular walls. So these are red blood cells in the middle of the blood vessel, and then these are the walls. And when you zoom in, you see all those little bright spots. So when I say it was maybe a strategic mistake to look at the dementia cases, is now many people around the world are thinking these things cause dementia. But it could be because we know that dementia is a disease where the blood-brain barrier starts to fail. It could be that the brain is simply a more hospitable place for the uptake of plastics. And we have to be very cautious in this interpretation. And that brings me to chapter 3. The thing we see most when we do a complete digestion of the brain and we remove all the brain and we exquisitely remove everything else, we see these weird little flakes. And we've been spending the last year and a half trying to prove that these are polymer, and we think we have pretty good evidence. So Aaron Ederle was, he's a scientist at the National Institute of Occupational Safety and Health. He was actually one of the victims of reduction in force this last year, which is part of why we haven't published. It's sort of slowed us down for 9 months. But he worked with a team out in West Virginia and Pittsburgh to look at 3,000 of these little particles collected from 3 organs from 5 different subjects, and just measure the length and the width of each of these. And what he showed was that in the brain, the average was less than 200 nanometers long and less than 50 nanometers wide. But the brain, on average, had bigger particles in it than the kidney or the liver. And this is a scatter plot of the length and the width. And basically what it's showing is the orange dots way out here are the brain. So it's not so much that there's different particles, there's just more big particles left in the brain, which is weird. But the brain isn't really an organ of clearance. Livers and kidneys are designed to get toxins out of the body. So I think they're doing their job, or at least as best they can. But the brain, it's kind of weak in this regard. So we did some spectroscopy. We showed that we think that if you look at the little trace, the red line is a pure polyethylene standard, and the black line is what our little flakes are showing. There's a lot of continuity there. We think these are polyethylene mostly. We did this study where we heated these things under an electron microscope, and you can see they start as these jagged things, but you start heating them, and they slowly like mold and fuse together as they melt. So this is where you have to imagine being a chemist. I'm not a chemist, I'm a toxicologist, so this has taken some deep thinking on my part. Sorry, organic chemistry comes in here. Carbon is the building block of these polymers, and so polyethylene is literally carbon, 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 6,000 carbons long, 10,000 carbons long, sometimes 600 carbons long. You know, it's random chain lengths. But there are regions of it where it just sort of folds back and forth, back and forth, back and forth, super tight, super dense, super tight. And then these little regions are connected together by less dense amorphous connections. So these are so tight that the chemicals that degrade this, like oxygen and hydroxyl radicals and all these things, they can't fit in there. So the connectors are what break apart. So these really, really dense regions, and this is like the supramolecular structure of you get these spherolite crystals, and they just look like little stabby anemone when you look at the very, very nanoscale. And this guy, Sanat Kumar, has been a collaborator recently, and he published this 2 years ago, last year. And he's saying, yes, when I just let plastic sit and I look really closely, I start seeing an emergence of this size range from like 5 to 500 nanometers. And they look like these little flakes that you're seeing in the human brain. So we sent that in over a year ago for review in Nature. And it was rejected soundly, and the reviewers didn't say we were wrong. They said they just don't believe us. And the big issue they had is because we're using this aggressive digestion method, we were creating these little flakes, calcium lipid crystals. Okay, so the reviewers suggested like umpteen different chemical analyses we should do to prove it, but I came up with a better solution. Working with our museum, I don't know, this is an older crowd, so maybe not, but they're a little older than me. There's a Museum of Southwestern Biology over on main campus at UNM. They have 600,000 samples of different animals that mostly collected in the Southwest, but collected all over. And so this young woman, who's now Dr. Eve Rowland, she graduated last year, she found this jar of kangaroo rats that were collected up in Rio Rancho. And they've been sitting in a glass jar, so no plastic contamination, since 1966. We got her to go back out. She had the geocode exactly. She went exactly to where they were collected, and there's now a taco cabana. So she went a little south and down in the arroyo and collected another 20 or so rats for comparison. We had the same recipe for fixation, so we know that we handled them and fixed them the same way. She pulled the brains out, and the brains were essentially the same size the 2 years, but when she digested them, you can see 1966 brains digest to a very, very small pellet. The 2025 brains, there's something inside them. There's something else there that doesn't digest. And when she's taken 1,358 images now from 10 rats, essentially, just trying to show exquisitely well, we just don't see anything. So these are 1966, and there are things, little mineral nanocrystals or residue from the digestion process, but over and over again we see these just massive piles of shards, these little nanocrystals in the kangaroo rat brains. Statistically, it's massively significant. Now, this is the most chemistry you're going to get today. I promise I won't do anything worse than this. We work with Sanat Kumar at Columbia again, and his postdoc Shrishti. They use this. It's an analysis called wide-angle X-ray scattering, and essentially it looks at the nature of the crystals in a sample. And what she showed was that in the human brain, you get these little peaks, and they're very tight, very normal. And then in the kangaroo rat from 2025, you get almost identical peaks, even more intensely. But in the 1966 rat, you get nothing. And so this is a control standard for polyethylene. You'll see that it's well represented here. And then this is polypropylene, and over here you've got your different kinds of polypropylene. They do line up at those peaks. We also did PET, PET. So, oh yeah, I skipped over some stuff. So we're very convinced these little crystals are the most durable parts of plastics that reside in the environment. These are kangaroo rats. They're not eating from our packaged food. They're eating whatever is in the environment. So and there's just so much of this stuff that's easily found in their brains. One thing I like to note is that because we do such an extensive digestion job, what we see under a microscope might not be what it looks like in place in the brain or in the kidney. And one of the things that's sort of been hypothesized is these little parallel regions, they break apart, but they probably stay really close because of all the organic degraded material, and they agglomerate, they accumulate together. And when we don't do as thorough a job, we do see these objects that look like they're stuck together flakes. So they may be bigger than these little, than like 200 nanometers, but they're composed of these little materials. And I won't go into this so much. This is kind of the idea that it's a very complex chemistry because you've got sort of these internal crystals, but they're surrounded by that aging, degraded polymer. So again, think about the dashboard of your car, or if you've ever had like a plastic lawn chair and after decades or even 5 years, the sunlight has caused so you can scrape gross stuff off of the surface of that. Well, all these will have a layer around them that used to be polymer and is slowly eroding away. But when they're inside the body, they're going to be wrapped by lipids and proteins from our body. So you may have increasing levels of complexity of those little shards inside our body, and that may impact a number of things, including how the body responds to it. They could be completely encapsulated, so your immune cells don't even recognize that they're there. We're still working on this. All right, sort of the last part of the story goes back to Hawaii. What we realized was that it's very, very hard to create micro or nanoplastics in the lab because fresh plastics are they're so pliable, they're so resilient and strong. They're great, they really are great materials. I mean, you can do so much with them. So what we did was we went out to Hawaii and brought back about 50 pounds of old aged plastics from this cleanup. And we grind them in a cryo mill and we wash them very well. And ultimately we're at a point now where we can generate kilograms of micro and nanoplastics for use in the lab. We're feeding these to mice, we're putting them on cells. We've shared them with 20 labs around the planet. Netherlands and Korea and Harvard and Baylor. They've all got a little of this stuff now, and they're finding that these environmental plastics are definitely more toxic or biointeractive than the little pristine plastics that you buy from the companies for laboratory use. So one of the things that's been cool about this study is we fed mice admittedly a lot of this plastic for 12 weeks. They liked it, they didn't lose weight, they thrived, they did well. And this is the size distribution. So only from 1 micrometer, 10, 100, there's definitely a lot of large plastics in our mixture, but about 1% of the mass is below 1 micrometer and therefore can be absorbed into the animal. After 12 weeks, when we looked at how much was in the brain, liver, and kidney of the mice, it clearly goes to the brain. 2 or 3 times greater concentration in the brain, and the kidney and liver don't really significantly change, maybe a little bit. I love it because we've been beaten up in the press a lot over the last year, and this is the most reliable evidence that we have that we're right. The other thing I'll point out is the pyrolysis GC mass spec is really tight in mouse studies. It's not like several magnitudes different. So in those humans, I think humans do really have largely different uptake and distribution. There really is big differences from human. I share this phrase with my students. All models are wrong in the lab. Some models are informative. And so people like to criticize mouse models, but what I will say is that humans are horrible models for other humans. There's just too much difference between us all. All right, so I'll wrap it up here. I'm at this point where we are 100% convinced that the plastics are in our bodies and they're going up over time. The levels in the brain were higher than expected. They seem to be higher than any other organ that we've measured so far. They're definitely higher in dementia samples, and we're not sure whether that's a chicken or egg phenomenon. The plastics we shared with a researcher at Harvard, Do-Yeon Kim, he's using these, we call them organoids, where you take samples of brain from humans. I didn't really ask him which humans. I probably need to get more information on that. But he had samples from neurologically normal brains and those from dementia brains. And he puts our plastics on them. The normal brains don't care. They're fine. Put all you want. We're going to be fine. But the dementia brains express all the bad things in a dose-dependent manner that you would worry about in dementia. So it may be that the plastics don't cause dementia, but it really makes it worse. So we're working through the nature of this interaction now. And that environmental nanoplastics, in some sense they're very conjectural because nobody's really proven that they exist and shown images, and we're really the first group in the planet to be doing this. But what we've concluded is that they're the nanocrystal regions from highly crystalline plastics. That's going to be polyethylene terephthalate, or PET, and polypropylene. Those are the major classes that will have these. PVC should not have crystalline regions. Polyurethane, polystyrene, they won't have that. So, but knowing that that's the problem, knowing that that's the material that does get into our system, now we can go back to industry without having to shake our fist and just say, look, start replacing these things, or figure out a way to incinerate them so that they don't just reside in landfills and get back into our irrigation system and ultimately into our crops and our food. So we're moving in a way that we're not just trying to rattle cages, we're really trying to create change to create actual change, not just have people lose their heads. The fun part about what I do is that it takes an army, it takes a village, so many great people, mostly at UNM, but NIOSH, Oklahoma State, we've got partners all over now, and I will brag that what we've done has made UNM like ground zero for this work, and it's really exciting to know that our university, because of sort of we're small, but we all work together really well compared to a lot of other bigger universities. We're not siloed. We've been able to get several grants for early career investigators. Alicio and Shang have brought in their grants. We've brought a lot of attention to this story, and this sort of builds from this. I give Will Stone credit at NPR. He came up with this wonderful title for the article about the plastics in the testes, plastic junk. I got to meet with Katie Couric, who's an absolute legend. I love her. A number of people have mentioned our work in popular media, but it culminated 3 weeks ago in a meeting in Washington, D.C., where the EPA and a group called ARPA-H, which is under the Department of Health and Human Services, announced that they're putting $144 million towards research on this topic. Whether we get it or not, I know that it will advance the field, it'll advance our understanding, it's a huge plus. The EPA has moved microplastics finally to a regulatory category. It is admittedly the first bottom rung of a very steep ladder to doing anything about it. But I'm not going to, I'm being skeptical, not cynical. And ultimately, we brought a lot of really positive press to the university. If you're familiar with how press works, the university never gets positive press. It's always like somebody's being sued, there's the hospital, and there's some, yeah. So it's been a real plus. And again, it's across campus. So it's engineering, it's biology, it's nursing and pharmacy and cancer center. We all work really well together, and it's been a blast being at this in this institute. So I will stop, and we can chat, ask questions. Thank you. Yeah.
If one of the challenges in trying to decide whether these things are causing dementia or another possibility, ADHD and autism, the fact that you can find very few humans living next to each other, where one has been exposed to plastics and others have not.
It's a massive challenge. The problem is we're all exposed to some level, so we have to look at gradients and make associative conclusions, which is not, I'm a rat and mouse kind of guy. I like to know that this was an exposure and this animal didn't get it. So this world we live in now, where we really have to do the human work at the same time, it makes things complicated. To your point about autism, such a political minefield right now. But if you were to ask me what, like a little inert plastic particle of that size and that frequency concentration in the brain, what could it do? It could interfere with the communication between cells. And that's the fundamental issue with autism, is the cells are not connecting very well, or they're doing it abnormally or whatever. So I really want to move in that direction in our lab, but I'm afraid to write that grant. So, yeah.
Should we be wearing masks around town?
I don't see that there's much in the air. We've done a very small sampling of what's in the air, and it's less than 1%, maybe 0.4% of all the particles you're going to breathe. So there's still, like diesel particles are 25% in this town because of the highway. Dust and other fragments of very small rocks is going to be another 20%, and then other things. So diesel particles are way more toxic. My previous life, and the reason I come into this microplastics world with a lot of skepticism, we used to use microspheres of polyethylene and polystyrene as sort of a negative control. This is what a particle does, not much. This is what a diesel particle does. Woo, the cells light up with all kinds of inflammation. So given how low, like I wouldn't worry about masks. This time of year with a pollen, sure, much more impactful to your health. We really think it's in the food. Yeah.
Yeah, so that's my second question is, back to that business of fat in the blood and the frontal cortex. Are we talking about the RFK diet?
You know, if you talk enough, some of the things you say might be true. So here, these are our data. Now these were, here it is. Admittedly, these were done with high school students and undergrads during a summer program. So we're not ready to publish these for a number of reasons. We're going to redo them and get better. But this is nylon measurements, and this is polypropylene, and we're talking chicken, venison, deli ham, grass-fed beef. The red bar in both of these is whale blubber. So really, really low, moving to beef jerky, beef stick, processed pork, which is Spam, vegan burger, the Impossible Burgers. So we're looking at this data. For one, I mean, UNM has like 3 lawyers. We can't publish this. I don't even know the heartache that's going to happen when Impossible Burger comes after us. But it's the processed meats. It's absolutely the processed. Vegetables are down here. We're starting to flesh this out, and we're trying to build a larger matrix to publish. But if you do eat meat, and you can stick to harvested venison or elk, or you can know that your supplier is. I hate using the word organic because that's a greenwashing label that applies to too many things. But if you know the source and they're raised in a healthy way, they should have low levels. But what I worry about is we're biomagnifying this stuff in our diet. So do you know how we treat our wastewater in our town as well as most other towns? There's a substance called sludge, wastewater sludge. So the water comes out and it basically mixes with sludge, and that sludge is really great at removing the microplastics. So the discharge that we put back into the Rio Grande or back into the Hudson Bay or wherever is relatively clean of microplastics. Problem is we take that sludge, we dry it out, and we use it as fertilizer on 60% of the agricultural regions of this country. So we are just focused, we are taking all the stuff that comes out of our urine. Our bodies did a great job. We're just putting it right back in. So I think we've got some fundamental problems that they're going to be. You can't just stop using sludge. We won't have crops, right? So there's things we do as a planet that are going to require some looking at. Oh, you're going to have to fight, Paul.
How have you changed your diet or plastics use?
How have I changed my diet? I don't eat as much meat as I used to. I've had 2 major changes in the last year. I've been taking a GLP-1, and so I don't eat as much. That's great. And my kids have gone off to college, so I'm a empty nester with my wife, and so our diet has gotten much healthier in the last year. And I promise it doesn't really, the plastics didn't really impact that. So yeah, but eat meat for flavor, don't eat it for mass. That's sort of our perspective on this. Enjoy good fruits and vegetables as much as you can. Yeah. Let me go to this in the back. Yeah.
Is the water we're drinking adding to the problem, or are they removing nanoplastics from it?
You can find plastics and microplastics, nanoplastics in the drinking water. I don't think it's a big concentration. It's kind of the argument about the air. Compared to the vegan burgers and the beef sticks, those are the big exposures. The water's probably fine. All the studies with the bottled water, typically, I mean, there's good evidence that there are some plastics that shed off of the fresh bottles, but most of the problem they have is they don't know what was in the water when it was bottled from the tap. There was a big paper a couple years ago from my colleagues at Columbia. It was in Proceedings of the National Academies of Sciences. It got a lot of attention. They're like, there's hundreds of thousands of plastic particles in bottled water. And the point of that study was not to make a fuss about that. It was really to say, we've got this new technology. It's called stimulated Raman spectroscopy, and it works so well. We look at all these things we can detect in otherwise really, really clean water. They didn't have that control of where the water was bottled from. They could have just reached, the Hudson River, do something like local. So they put out this big paper, got a lot of press, and then a lot of people came and attacked them, and it's like, that's not the point. There's really relatively low concentrations in the water. Sorry.
I'm curious what your opinion on biodegradable plastics is hastening the problem, or?
Skepticism is warranted. There's sort of a mantra now from the world of plastics researching is like, no testing, no market. If you don't confirm that your new material is going to degrade, it's not going to release plasticizers, you as Chevron or Dow, you guys have more money than God, you should be testing all of this. So we're trying to make that a reality, but of course globally, there's this global plastics treaty from the United Nations that hasn't gone anywhere. It's a big mess. I think federal legislation is the way to go. I don't think local legislation really makes a difference. We ban plastic bags and pat ourselves on the back, and it's trivial impact. So,
So I'm just curious, is it a good idea for the stuff to break down, or is it a bad idea? Should they be actually doing the opposite and trying to make the plastics hang around longer for pieces?
Right now my thinking is about the crystal structure. If it doesn't form that crystal structure, it should be okay. I do like the idea that they break down faster. And essentially the component of those biopolymers or the biodegradable is polyethylene is carbon, carbon, carbon. The bond between carbons is really quite strong. If every now and then you poke an oxygen in between them, that'll cause a break. And if you poke it in frequently enough, you won't get long chain carbons, you'll get relatively short chain that are easily used by microbes or plants. So my whole thing is return to the carbon cycle, right? Become CO2 or something that can be utilized. I think I'm optimistic that the biopolymers tend to do that, but I agree the companies who make them have to find out. Yeah. Go ahead.
So I've read occasionally about some research on trying to break the carbon cycle in plastics and different fungus and other chemical processes. Where are we at with that?
I mean, people are trying to use all kinds of cool things to degrade these faster using, if you think about the world of venture capital, this is the new gold rush. Trying to engineer a microbe that will degrade plastics right now is like...
Is there any hope on that?
My biggest fear in this whole thing is human cleverness. And so you think about like every zombie movie you've ever seen. You kind of start with this principle of like, hey, we've got this new microbe and we're gonna, next thing you know, it gets in your stomach and everybody's free of plastics but dies of dysentery. So it comes back to the biopolymer clause. Like, let's make sure that these are, actionable, sustainable, inexpensive, all those things.
Okay. And that was my reaction to reading.
Yeah.
So that's why I was asking.
I've been talking to people in industry more and more to get a feel for what the real depth of the problem is. And there's this company called Atlantic Packaging. Obviously, they make $1 billion a year by creating materials for packaging, and most of it is traditional polyethylene related stuff, and they're trying to move to biodegradable. It's 12 times as expensive. Even if you were to increase the bulk of material, you could only trim that down to maybe 6 times as expensive. And so the only companies that are really willing to move in that direction are the companies where it's a sales pitch to their customer base. So your Patagonias, your Trader Joe's maybe. So you start thinking that they will make a difference, but the fast fashion we get from Amazon, they're never going to care. They're not going to bear that cost. So, yeah. Go ahead.
Oh, I'm just curious what you mean by vegan burger. I've been eating a lot of like the Impossible fake meats. Is that a term that includes all of them?
I would assume so. There's Beyond Burger and Impossible Burger, and I'm forbidden to know which one was which. It was one of them. This one out here where the bacon is really high, I actually think that's an outlier. The other two samples were quite low because bacon's not processed. It's smoked. It can be taken, harvested from a healthy situation from the pig, and but I think these processed things, every time you're grinding, you're adding glues, and you're, whatever they do to make these things, those are the opportunities for contamination. The black bean burgers should be okay. I don't know. I haven't tested those yet. Yeah.
Yeah, just I hope I can say these correctly. A couple of questions. There's been some thought about how good the analysis is using gas chromatography and mass spectrometry. And is there any thoughts or any methods being developed that might also verify the results that you have?
Yeah.
And then the second question, well, let me just state that one. I guess the second question would be, is there any kind of diagnostic imaging using neuropeptides and PET scanning, this type of thing, that might reflect the amount of disease in live human brains with regard to plastic accumulation?
All right, two good questions. The first one is the reason we've gotten a lot of bad press over the last 4 or 5 months is there is a letter to the editor in the Nature Medicine, which I actually encouraged it because I thought we would have better discussion around the need for better methods. And it's been fine. It's fine. Everything's fine. But the principal argument is that when we do our digestions and we do this pyrolysis, we're looking at this long chain of carbon. Well, lipids in your body are long chains of carbon. And so there's this concern that like fats might be contaminating our samples, and that's what we're measuring. And the idiot from Germany, who's an analytical chemist and has no idea about biology, was posting on social media, brains are 60% fat, kidneys are 5% fat. Well, that's comparing wet weight to dry weight. Start there. So if you completely desiccate the tissue, the brains are 60% fat. But if you completely desiccate kidneys, they're 20% fat. It's the cell membranes that are phospholipid, right? But here's my answer to that. So on this scale, I don't have the brains represented here, but polypropylene was around 500 in the human brains, so about with vegan burger. See this nice one here, whale blubber? 80% fat, dry weight. So it's not just fat. Same thing with nylon, same thing with polyethylene. So we're more right than we're wrong. The speed of light is a known number now, right? 3.8 times 10 to the whatever. Galileo hypothesized there was a speed of light, but he couldn't figure out what it was. And then 150 years later, some other scientist came along and we tested it. It is 2.8 times 10 to the 8th meters per second. And that was wrong, but it lasted for 50 or 60 years before somebody came along with a better test. And we eventually got closer to 2.998 or something. That's where we are. It's a young science. I don't think this isn't a physical constant. There is no specific number that all human brains will be of plastic. There's going to be a lot of variation. It's going to depend on whether you're using acid or base to digest it, depending on how you process through tubes. So we're working on that, and we're talking with a lot of different labs to figure out best methods. Second point is about, is there a PET scan or a MRI? We're working with 3 groups right now to figure out the MRI, and I'm not optimistic. They just look too much like fats, and they're not that big. But the group in the Netherlands showed that if you use susceptibility-weighted imaging, the environmental plastics, because they've been out there, they've absorbed some ions like calcium and magnesium. They might have a magnetic dipole that can be detected at a high enough concentration. So we're working on it.
Yeah, okay,
Yeah,
great.
yeah, we're working on it.
Thank you.
Margaret?
Well, you hear pretty often here now, Albuquerque people say, oh, the city doesn't really recycle the plastic. Do you know whether the city really recycles the plastic or not?
I want to say they do, and nobody should change their behavior, because there's no going back if we stop. It's more about managing humans than about finding a truth. What I do know is that those stupid plastic bags, those grocery bags, they cause about, I've heard they cause $6 million of loss for our waste facility, because they get thrown away and they end up getting tangled in the compressors for garbage. And they tangle up, and there's downtime where they have to go and clean those stupid grocery bags out. And it ends up being like a $6 million loss for Albuquerque. So if people could just carry their darn reusable bags, we'd save a lot of money right there.
Speaking on that, why we had it here briefly before the pandemic, a plastic bag ban, and now that seems to, Mayor Keller doesn't want to do that anymore.
Pandemic came along, and they were like, just we don't care anymore. Just don't breathe on anybody. So yeah, we did that, and we, like California, moved to those slightly thicker, reusable bags, but people just still treated, because they were free, people treated them like they were throwaway. And we have data from California that they actually quadrupled the amount of plastic waste as a result of that ban. So that's why I say, like, we need a federal regulatory structure like the Clean Air Act to start addressing this, because my colleagues lost their mind when Trump came back into the office, and he's like, we're getting rid of this plastic straw ban. And they're like, no, why would he do that? And I'm like, because that was kind of a punk move by Biden to do nothing and claim a victory. Yeah, it's plastic straws. That's not really a big part of the problem. So yeah, the local, I've helped Santa Fe push for their regulatory changes, and the problem is we just need Lee Zeldin, who's an administrator for the EPA. He needs to make a regulatory structure, get Congress to pass it. We'll see what happens in November. Yeah. Oh, sorry.
Recognizing this is a young science and we really don't know the risks, is there any speculation you speculated a little bit about autism, but any experts that have any rational reason to suspect one thing or another might be a consequence?
The cardiovascular thing is coming out as probably real. The initial study from the Italian group showed us a really high risk, probably, we're starting to see in animal studies. Yeah, you treat animals with plastic particles, there is worsened atherosclerosis. Ross Clark here, he's a vascular surgeon, he's been doing a lot of work in this area, and corroborating what the Italian group did. The mouse studies are controlled exposures. We're starting to get a better picture on that. We're now doing a dementia model. We're working with these dementia researchers, so give us a year or two and we'll start to have a little more understanding. I look at what diseases are going up in frequency in the world. There are not many. And it's funny because a lot of the current political movement is from the Make America Healthy Again group, and if you listen to them, they're not based in fact. They will say things, skyrocketing cardiovascular disease. Cardiovascular disease is about the same as it was like 10 years ago. It's not changing that much. Dementia is getting higher because we live older. You know, so there are some diseases that are getting worse over time. Globally, multiple sclerosis is getting worse. Parkinson's disease. Colon cancer in young people. Under the age of 50, there's been a steady rise in colon cancer since 1994. Early onset, ER-positive breast cancer is happening in younger women, and it's more aggressive and more devastating. Along with the colorectal cancer, appendiceal cancer, so cancer of the appendix. Very low incidence. You might not ever have met anybody who ever has it, but it's doing this right now. And I'll bet the mechanism is very similar to why colorectal is starting to trend up. Male...
From other pollutants other than plastic?
Could be. Yeah, so all the other plasticizers that go along with this. I don't think that the plastic particles are carrying these with them, but they already put them, they released them and they left them in the environment. So they do tend to traffic together over time. So it could be phthalates and bisphenols and PFAS and things like that. Yeah. Yeah. Terry?
Oh, yeah, just I just read a report in just yesterday, and but it mentioned that exposure to pesticides might be related to the increased incidence of colon cancer in younger people. And I thought that was interesting. One of the thoughts that I had was, I think the dementia issue is something that I'm certainly interested in. But I don't know how to separate out if plastics are a causative factor or the result of say, reduced particle control in the brain, in the frontal lobes when they in fact become demented. In other words, the blood-brain barrier kind of breaks a little bit and that kind of thing.
Yeah.
Do you have any thoughts on that? That's why I was looking. Is there any alternative ways of looking at this to see?
Yeah, actually, I get a little philosophical, and it's, we talk about pathology as if that's the disease. Pathology is the step in the path to the disease, especially in dementia, where a lot of people have high levels of these tau and amyloid beta plaques, but they don't have cognitive issues. So, there may be something where there's an interaction between the pathways, biochemical pathways that are, I don't know, but we're working with.
Yeah, you're in the right field, that's for sure. That's great. I love it. This is terrific.
Yeah. So, did you want to ask one? Oh, Charlie? Yeah.
A lot of us live in rural areas where we're on well water.
Yep.
And I wonder, that well water, of course, gets recycled, filtered down through the earthen filters. Have there been any studies about the difference in plastic concentration in well water than, say, city sources?
There must be, but I can't think of any right now. It should be cleaner, theoretically. My concern would be, is there a local landfill that would be contributing to your groundwater supply? And if there is, then I'd be concerned. And of course, the problem with groundwater is you don't know all the places it goes to. Kind of like when you have a roof leak and the roof is leaking here, but the hole is over here somehow, and it just sort of trends down. But I would think there's nothing specifically to worry about for the well water with microplastics.
And on the other hand, what is the component of vegan burgers that puts them at such a high concentration?
They're so heavily processed, so there's glues and other ingredients that are put in there for texture. So I'm not an expert in the food processing world, but figuring out what are the steps for contamination, I think is going to, I don't know, we have limited bandwidth. So trying to become food processing scientists might be overstretching our ability. It's really curious, though. Yeah. Yeah.
I noticed that you have a lot of meats, but I don't see any seafood or any fish out there. Have you done studies on that?
Whale blubber. They're not a fish. That's fine. We haven't. What I will argue is that we've had this story backwards, because these plastics were discovered in the ocean by the oceanographers, and bless their hearts, I think like Richard Thompson, Marcus Ericsson, Chelsea Rockman, these are, I mean, I hope there are statues made to these wonderful people for their seminal discoveries. But the plastics don't start there. They start right here, and they leak out to the ocean. So I think they were just exceptionally, like, the ability to find something like that is really amazing, just so perceptive. But the story really is what we've contaminated here underneath our own feet. That's my fear. And people are just now starting to look at that. So, somebody in the back had a question. Yeah.
Yeah, I was curious about the ability of the brain to filter out these things, and the glial cells. Is there some implications for how that functions differently than, say, the kidney?
The brain is not a great filtering and removal kind of organ. It's designed to pull things in and use them. And recycle. And we're studying that now. Elian El-Hayek is, I should say Elian El-Hayek is the one who took that original picture of those little crystal flakes. And she's the one who brought that to our attention as, look, I just see these. I don't know what. So in this whole story, she's the Rosalind Franklin. She's the one who took that image, and all the science that arises from that. We need to make sure Elian El-Hayek gets full credit for that perceptive observation. She's working with CSF. She's working with us, University of Virginia, University of Iowa, neurologists, like all different kinds of syndromes where you can get cerebrospinal fluid. It's a weird wide range of, like, there's children who have hydrocephaly. There's, dementia patients. There's all kinds of injury cases where you need to clear the fluid to reduce pressure. So she's working on that. Multiple sclerosis is another one. So give us a couple years.
Yeah. Okay.
Yeah.
And it's also in the cerebrospinal fluid, not just the brain tissue.
Yeah, and in fact, we had this great meeting in Santa Fe back in January where people all over the country and a couple other countries came, and Columbia, University of Columbia, the same group that did the plastics water bottle study that got so much attention, they're doing CSF with their stimulated Raman. Their data completely overlaps with our data, and they're showing, like, the average human would have 100 million of these little fragments in their CSF. I mean, they're like viruses, so they're very small, but it's a lot. Yeah. Yeah. Mm-hmm. Yeah.
I noticed you have deli ham and processed pork. I thought deli ham was processed pork. So there's obviously a difference.
The deli ham is still like an intact ham, and it was smoked. But the processed pork is short. It's us not using the trademark name of Spam. Yeah.
So don't worry about. I'm just exaggerating a little bit. Don't worry about bottled water, okay, so much as the diesel or the secondary exposure and cycle. So what would you guess might be the percentages of personal contamination to a human in a lifetime of the primary first cycle through versus the secondary and eternal cycles?
Yeah, I mean, I think most of the problem, like 80% of the problem, is the secondary generation from old, degraded, thrown away plastics that gets into our food. And so I think it really is meats. I think it really is second tier is going to be vegetables and maybe a log difference, tenfold difference between them. Vegetables are going to be much lower. And then the water contributes, and then the air. And I honestly think there's that tenfold difference in exposure across all of those, from meats to vegetables to water to air. You think about the mass of particulates that you breathe in, you're talking nanograms versus nanograms, micrograms, milligrams, or grams. That's 1,000, 1,000,000, 1,000,000,000 times greater from the food. So I really think that that's the principal concern. Yeah.
Are all plastics petroleum in origin?
Well, no, to the point about biopolymers, there's new materials that are being used to generate these, and they can come from plants and cellulose. And of course, if it does come from cellulose or hemp or something like that, there are thousands, 10,000s of years of evolution on this planet to turn those back to the carbon cycle. But these polymers from petrochemicals, that no organism has had a chance to figure out what to do with these for energy or other reasons. Yeah.
And the so-called forever plastics are in what category? Are they petroleum, are they biofilms, are they?
So the forever chemicals are plastic additives, PFAS, so perfluorocarbon, there's different types. Those are really rigid molecules, so not particles, but they're molecules, and they don't go away for a long, long time. But on the other hand, because they're so rigid as molecules, they don't really interact with our body that much. They're very stable. So you have to have very high levels to cause any biological effect, and we're still struggling with this because the chemical industry is like, no, we did the testing, there's nothing there. And we're like, yeah, but it's building up in the environment because you keep using it and it doesn't go away. But those are fluoride bonds, the really strong compared to carbon, so it's really hard to break those. Yeah. Yeah.
So first, I'd like to thank you for what you're doing.
Sure thing. Sure. Thanks. They pay me fine.
Yeah, but you chose this career. So I stopped washing plastics in my dishwasher. It sounds like it's not important.
Probably doesn't matter too much.
Yeah.
And in fact, once you've washed it like 10 times, you're probably not getting more things off of it. Yeah.
So what would you like us to do?
Enjoy your life while you have that.
Shake your fist at the man. I don't know. I worry that I'm shaking my fist at the man. Vote at the. Yeah, let's change the mentality of leadership in this country to get them thinking about long-term solutions. I like that, and I could even get emotional about representation from our state is really good at the federal level. Melanie Stansberry, Ben Ray Lujan, Deb Haaland, these are people who think with a longer term mentality, and we need a little bit of that. Too much stuff is built around quarterly profits, and I don't know where all that leads us. What do we do? Don't buy junk. Don't. The little trivial trinkets and gifts we give to everybody, like the little McDonald's toys in a box. Those things are just, they're landfill before they're anything else. Those are the things that bug me. Yeah. All right. Thank you all. This is a great discussion.